In order to maintain homeostasis, the body must constantly resist infection by pathogens. There are two general types of disease resistance: nonspecific and specific. Nonspecific resistance consists of defense mechanisms that are present at birth. They act rapidly, but provide only general protection against infections. These mechanisms include barriers, antimicrobial substances, phagocytosis, inflammation, and fever. Specific resistance is known as immunity. Such defense mechanisms develop in response to specific foreign cells or substances called antigens. Immunity needs time to build up each time a new antigen exposure occurs, but eventually acts very effectively on that particular invader. Immune responses triggered by antigens include cell-mediated immunity and antibody-mediated immunity. Both types of resistance mechanisms work through the functions of a group of antimicrobial substances called complement proteins. Once physical barriers, such as the skin, are breached by pathogens, other resistance mechanisms mobilize. Cells, such as macrophages, phagocytize pathogens. One function of these cells is to increase exposure of antigens to immune cells. Such exposure begins the activation of immune cells and leads to the production of antibodies. Antibodies and antigens bind to form antigen-antibody complexes. In turn, these complexes bind with complement proteins to trigger a cascade of new complement proteins. The entire group of complement proteins functions to enhance phagocytosis, cytolysis, and inflammation. As the immune response is enhanced, antigens are made more available to immune cells, which produce even more antibodies. Antibodies bind to antigens, forming even more complement proteins. This results in the activation of more and more complement proteins, and eventually produces a fully activated immune response. For a pathogen to enter the body, it must penetrate the physical, chemical, and biological barriers in the skin and mucous membranes. Once the pathogen enters the tissues or blood, it encounters macrophages that carry out phagocytosis and natural killer cells that execute cytolysis. This initial encounter can trigger the production of chemicals that promote fever, inflammation, and more phagocytosis and cytolysis. An infection can hold out against nonspecific mechanisms and even overwhelm this initial resistance. Meanwhile, immune cells are being exposed to the antigens, initiating a specific response to the invader. Activated B lymphocytes produce antibodies, and activated T lymphocytes secrete chemicals to lyse or burst the infected cell. In addition, activated complement proteins enhance phagocytosis, lysis of cells, and inflammation. The processes of initial specific resistance result in an increase of the number of available antigens and trigger the proliferation of B and T cells. The number of antibodies increases exponentially. After a week or so, the specific responses effectively destroy the infecting microbe. Specific resistance remembers how to fight antigens that have triggered the immune response in the past. This is known as immunological memory, and for particular antigens can last for decades. Prior to the first exposure to an antigen, levels of antibodies and lymphocytes are low. While immune cells fight the first exposure, long-lasting antibodies and long-lived lymphocytes, known as memory B and T cells, are produced. The high number of memory cells produces a faster and more effective response to subsequent exposures to the same antigen. Immunological memory is the basis for immunization vaccinations, in which a small, measured exposure to specific antigens enables a secondary response against future exposures to the same antigen.